today I'm going to talk about physical mechanisms of selection. This will be somewhat of a review presentation. I'll put together information that's come out of quite a few different labs. But the idea of this is not so much to try and explain how selection happens in one particular case or another, but rather to talk about some of those basic physical concepts that can lead to selection. So we've got a toolkit that we can use to understand selectivity in a range of different situations. So first of all, we might ask, why do we care about the mechanisms of ion selectivity at all? And I probably don't have to convince this audience too much, but I can think of quite a few different reasons. So in biology, we know that ion selection is critical for cellular function. So just as a couple of examples, we know that potassium channels have to be able to block sodium while allowing a large current of potassium ions in order to set and maintain the resting potential of cells. In fact, the potential that can be achieved is related to that selectivity. And they have typically 100 to 1,000-fold selection for potassium over sodium. Another example I'll mention today is that of calcium channels. So we know they have a role in converting electrical signals to cellular responses, but to do so effectively, they need to prevent the passage of sodium ions. And again, we have about 1,000-fold, up to 1,000-fold selection for calcium over sodium. But it's not just in biology, it's also in designing new technologies and industries in many different fields. So just as an example, people that are interested in water filtration and desalination are very interested in selectivity because they want to know how best to remove block ions while letting water pass through. And there are some subtle questions as well. So for example, why is it hard to remove some species from water, such as nitrates, while fluoride, which is quite a small ion, is comparatively easy? We might be interested in separation, for example, to get raw materials like lithium, to concentrate lithium as a material for battery technology, or to separate out radioactive ions in site remediation, or just as a separation technique for biomolecules. And of course, there's a lot of discussion around using these kinds of techniques for energy conversion and storage as well. Okay, so I said that I want to go back to the basics and the physical mechanisms involved in ion selection. And I want to put them into sort of three categories before I start going through them. So if an ion is to enter a narrow pore, and generally it has to be a narrow pore to obtain significant selection, then we know that it has to move from aqueous or a solvent into the narrow pore. So there has to be a desolvation or dehydration effect. And that can be different for different ions, yielding selection. The ion can interact with the pore, and if that's different between different ions, that can also yield selectivity. And then the ions can interact with each other. And so that gives you a sort of third category for how this can happen. So in fact, in my talk, I want to go through 8 different mechanisms that I can think of for achieving selection. And as I say, I'm not out here to try and say that one is particularly the most relevant in a particular case, although I will give some examples, but rather to talk about these as a kind of toolkit that we can use to understand selection in a range of different situations. So they range from sort of the background dehydration differences through to differences at specific sites, to the different kinetics and the competition of multiple ions. And we may think of these as distinct mechanisms. There might be situations where one is the real origin of selectivity. But another way to think of these is as different faces or different ways of looking at a general mechanism. So I can think of many cases, and I will give a few examples, like potassium channels in particular, where a good number of these 8 are probably at play simultaneously. They're really just different facets of one significant underlying process. Okay, so with that said, let's go on and start talking about what these different mechanisms are. So I'll roll the dice. I'll come up with the first mechanism I'm going to talk about, which is the simple dehydration energy differences of different ion types. So as we said at the start, if our ion is beginning in an aqueous environment, we know that it's likely to attract a shell of surrounding water molecules. So we think of this as the hydration shell. Now, these molecules may interchange from time to time, but of course, on average, we have this shell of water molecules attracted to the ion. And they're attracted there simply through the electrostatic, or primarily through the electrostatic interactions with that ion. But this attraction is different for different ions. So you're probably quite familiar with the fact that sodium holds onto its hydration shell more tightly than potassium. One way we can quantify that is with the hydration free energy. So this is how much energy is released when the ion is placed into water, and the magnitude is greater for sodium than it is for potassium. Conversely, we can say it takes more energy to remove those waters from sodium than from potassium. So if we're going to move these ions into a narrow pore, and if the pore is too small to allow the hydrated ions through, we have to dehydrate the ions, and that's going to yield some kind of energetic penalty, and there may well be a difference in this dehydration energy for those two different ions. So straight away we have an underlying selectivity for this pore, and this is without having to consider any specific ion-pore interactions. So let me give you an example of where this might be important, or where it's quite obvious, and that's in the selectivity in simple bare carbon nanotubes. So here I've used this essentially as a model of a non-polar pore where the ion has very limited sort of interactions with the pore itself. And therefore most of the differences relate to the dehydration effect. So what I plot above is the free energy required to move the different ions through this pore. And we can see that all these ions have a barrier to move through the pore, but this barrier is smaller for potassium than for sodium. And so here we might say that there's a small degree of selectivity for potassium over sodium. Potassium is going to find it easier to pass through this narrow pore. Okay, this sounds relatively straightforward, but I want to just point out that even this simple concept can yield quite surprising behavior. So one is that the selectivity actually depends upon the pore radius. So here's the example we just talked about. This is a very narrow pore, and a little bit counterintuitively, it's the big ion that's favored from what I just told you. It's potassium that finds it easier to pass through compared to sodium. And I should say I'm using this example of this comparison of potassium versus sodium a lot today, although a lot of these principles would apply to any ion pair you choose. Now, the reason for this is that the pore is narrow enough that both ion types have to dehydrate, and it's easier to dehydrate potassium than sodium. So provided the bare ion can fit in, this small pore is going to favor the bigger ion. But if we choose what I think of as a medium-sized pore, we actually get the reverse effect. So now the small ion is favored, sodium over potassium. Why is that? It's because this pore is now big enough to fit the smaller hydrated ion with its water molecules, but the larger potassium can't fit in with its surrounding hydration shell. And so in this case, sodium finds it easier to pass through than potassium. Obviously, there's a point in between where these balance out and we'll get no selection. And if we go to a wider pore, both ions fit through with the hydration shell, and we can effectively say there's no ion selectivity. In fact, if we just look at currents, that's going to be determined by the bulk ion mobility. And in this example, we'll always have just a very slight preference for potassium over sodium. Okay, and now before I go on to the complex cases, I just narrow carbon nanotube, and it does favor the larger potassium ion. A sodium ion is a medium-sized pore, like this medium nanotube, favoring sodium ions. But of course, in these complex biological cases, we also have complex interactions of the ion with the pore wall as well, and that's something that we'll go on to talk about in a moment. But it does sort of highlight that the dehydration effect is sort of a very critical underlying effect in all of these cases. Okay, so let's start to look at the more complex situations, and that's when we start to consider the interactions of the ion with the pore as it moves through. So in this case, there's always going to be this underlying dehydration penalty, but that can be compensated by favorable ion-pore interactions. And that's in general going to be quite important if we want to get large currents to overcome that dehydration penalty. And it's going to be the balance of the repulsive dehydration, the attractive interaction with the pore, and how that differs for the two different ion types that's going to yield the overall selection. Okay, so now before I go through some more of these different mechanisms, I do want to introduce the prime example that I'm going to talk about today, and it's one that we're all pretty familiar with, and that's the idea of selectivity in potassium channels. Now, my reason to do this is just to make sure we're all on the page with the structure before we talk about use this as an example to talk about some of the mechanisms. So as you know, potassium channels are made up of 4 protein subunits. There's a narrow selectivity filter at the top of the pore here. And if we zoom into that, what we find is that this narrow part of the pore is lined by backbone carbonyl groups and one side chain carbonyl. And they form essentially rings. So I'm only showing 2 of the 4 here, but there's a ring of 4 carbonyl groups, or rings of 4 carbonyl groups, and between these are quite well-defined potassium binding sites. And you can see these sites in X-ray structures of the protein. Okay, now I just want to have that in mind when I start to talk about some of the other mechanisms and use this example. Okay, so mechanism number 2 that I want to talk about is what's often thought of the snug fit model or the rigid cavity model. Now, this is quite an old idea. Besanier and Armstrong in the 70s, made popular again by Rod McKinnon when the first potassium channel structures came out. And the idea here is to think of the protein or the pore as a fairly rigid entity. And so if we look down the pore, what we would find in the case of a potassium channel is that there's these rings of carbonyl oxygen atoms. And in the X-ray structures, you could see that potassium ions fit very neatly inside these rings. What that means is that the potassium ion is nicely coordinated to those carbonyl oxygens. And so the interaction here can compensate for the dehydration penalty of moving out of water. However, if the pore is quite rigid and we put a smaller ion in, it can't form these favorable coordination distances. And so the interactions with these carbonyls is less able to compensate that dehydration cost. As a consequence, with this rigid cavity or this rigid pore, we think that potassium fits in better and is likely to be selected. And obviously you can get similar kinds of selection for different ions with different pore sizes. Okay, now this is a good mechanism, and I should say with all of the mechanisms I'm introducing, they're all physically good and plausible mechanisms that can hold in different cases. Whether or not any particular one of them is the one that's used in potassium channels or others is something that's open for debate, and I'll offer just a little bit of commentary. So one thing to keep in mind with protein pores is that proteins are generally quite flexible. So it's not necessarily easy to maintain the kind of rigid cavity that I've talked about in this mechanism. So if we look at structures of potassium channels in the absence of potassium, you'll find that the pore diameter does tend to decrease, suggesting flexibility. And simulations such as those by Benoit Roux do suggest that selectivity can be obtained without a rigid cavity. So maybe it's not the primary factor in potassium channels, but it's definitely a way to generate selection. Okay, so let's talk about the third mechanism, and this is really just a variation on that rigid cavity concept. So, and this is the idea of having a strained cavity. So here we might say, okay, the pore or the protein isn't necessarily rigid, but if it's going to change its shape to adapt to a different ion, that might come at some kind of cost. So we can imagine if the carbonyl oxygen atoms are going to move in around a sodium ion, there might be some kind of strain induced in the protein because of this. That creates, that comes at an energetic cost, disfavoring sodium inside the pore compared to potassium. And again, this makes complete sense. Any kind of strain in that protein is going to have an effect. Now, this strain can come from many causes, and I think it's actually a really challenging thing to quantify. So again, Roux and colleagues have came up with quite a good mechanism for trying to decompose this, but it's quite challenging because the strain can come from so many different causes. It can be just stretching of the carbonyl bond, which is sort of what I'm suggesting here. It can be, you know, very subtle conformational changes throughout the protein, or you know, pairs of atoms just becoming slightly closer or further apart. So it's I think we understand the concept of this mechanism quite well, but it still remains quite a challenging one to quantify effectively. Okay, now the fourth mechanism I want to introduce is the last of those that's related to the shape and structure of the pore or the cavity itself. And this is one that I call the reduced ligand fluctuation mechanism. And it's perhaps a little more subtle than the last two. But the basic idea here is that if we look at an ion in bulk solution, so imagine we've got potassium in water, if we look at the region that's sort of sampled by that water molecule around the potassium ion, we'll find that it actually moves about and jiggles about with thermal motion in quite a large volume. And so this brown shape is trying to represent that volume that's sampled by the coordinating ligand. Now, if we move that ion into the pore, here our ligand is a bit more constrained in how it can move. It's attached to the rest of the protein. There's perhaps less space and flexibility. And so that ligand may only be able to occupy a smaller volume or fluctuate around in a smaller volume. Okay, that's something we might expect, but if that difference in volume is different for different ion types, that change in volume differs between different ion types, then we actually have an entropic difference as we move different ion types into the pore, therefore a free energy difference and a mechanism of generating selectivity. And in general, any process that constrains the movement of ligands is probably going to favor smaller ions. This is quite a subtle effect, and I think it's unlikely it introduces very large degrees of selectivity. Okay, so all of those mechanisms were related to the shape and structure of the pore itself. Now what I want to go on to is a couple of mechanisms that are more related to the chemical nature of the pore. And so here, number 5 is one that we think of as the chemical nature of the ligands. And this, I think, is a really important concept to understand when talking about selectivity. So I want to start with perhaps a more obvious example. So if we have sodium initially in water and it moves inside a pore, but this is a pore that's highly charged, so we could imagine it has glutamate residues, carboxylate groups that line the pore, then we would imagine that sodium ion is going to be attracted into the pore. It's going to have strong electrostatic interactions with that pore, and it's going to like to enter. But now what if we compare it to a calcium ion? So calcium, divalent charge, it's going to be attracted into the pore even more. It's going to have stronger interactions, and so we might think if there's competition, the calcium is going to be the one that's going to enter. Now, which one enters is going to depend on the strength of the interaction between that ion and the coordinating ligands, and how that and the sort of repulsion between those coordinating ligands. It's the interactions between all of these atoms involved. Now, it can become, of course, much more subtle. So if we have sodium moving into a pore lined by carbonyls, like a potassium channel, is that going to be more favored or less favored than a potassium ion? And here it starts to become less obvious, but still quantifiable. And this concept actually goes back a long way. Eisenman pioneered this when looking at selectivity on electrodes, and then it's been applied by Akvist and then again Roux and his colleagues to explain biological selectivity. And there's a few different ways you can go about trying to quantify it as well. And what I'll show you is following on from Roux, using these very simple toy models, as we used to call them, to understand the nature of selectivity. So in this particular case, maybe potassium's the one that's favored. So how can we go about quantifying it? Well, this graph is just trying to give you, show you the concept of how this works. So the y-axis is showing the relative free energy. So anything above the dotted line is representing a situation in which potassium is favored in this pore or this binding site. Anything below the line is representing a condition in which sodium is favored. And then the x-axis here is just trying to do a simple way of changing the chemical nature of the coordinating ligand. So here we're representing it by the partial charge, or equivalently the dipole moment on that coordinating ligand. So if we have a very highly charged ligand, we're at this end, and we make it easier to favor sodium ions. If we have a kind of non-polar ligands, low dipole moment, it's easier to favor potassium ions. And I don't want you to worry too much about the different lines. They all show the same kind of trend. Now, this kind of behavior shows itself in biology a lot. We know that any binding site that is designed to favor something small like sodium almost always, dare I say, does always contain a fully charged ligand, a glutamate and a spartate at the binding site. Whereas if you have a binding site that's that favors potassium or a larger ion, it essentially never does. It will have carbonyls, it will have hydroxyls in that part of the protein. So this general behavior is perhaps a little bit familiar. Okay, we're whizzing through all these different mechanisms, and I'll try and put them together again towards the end. So mechanism number 6. Here, this is sort of an addition to the last one, where we're not going to look at just the chemical nature of the coordinating ligands, but we're going to look at the number of those coordinating ligands. So this graph is very similar. The top area is a condition that selects potassium. The bottom area is a condition that selects sodium. And what we've seen before is a low dipole moment ligands make it easier to select potassium, and ligands with a high dipole moment, represented at the bottom, make it easier to select sodium. But the number also matters. So if you have very few ligands in a binding site, it's going to be easier to put potassium in there. If an intermediate number might favor sodium, a large number might favor potassium. So to give an example, in the potassium channel, those classic binding sites I introduced before are between 2 rings of 4 carbonyls. So that's 8 ligands. And if there's not enough flexibility in the protein for those ligands to kind of flip out, move away from the pore, then you might think the number of the coordination number is somewhat limited. And so we might be at this 8-fold end which is going to favor potassium more than sodium, irrespective of the nature of those ligands. But you can see straight away from this, there is obviously a balance of the chemical nature and the number of these ligands. Why does 8 favor potassium? Well, essentially it's hard to fit 8 ligands at an optimal distance around a small ion. Okay, before I go on, I just want to highlight something that this was some work we did that I was quite proud of, and I just wanted to highlight this, how you can put together these different concepts to look at, how a range of different conditions can yield selectivity. So this is what we thought of as mapping different conditions, and it's just a variation on the graph I showed you before. So the x-axis is showing the number of ligands you've got present. The y-axis is the chemical nature of those ligands, and we were just trying to look at regions in sort of chemical space that would yield selection for different ion types. And I want to point out that there's this big black region, and this basically represents the situation where an ion is unlikely to want to leave water to enter that binding site. It would prefer to stay, thermodynamically would prefer to stay in the water. But then we get regions that favor sodium and potassium, and you can predict by how much. But I'll come to this in a moment. There's quite a few assumptions here that maybe limit this approach. Now we got a little bit perhaps overconfident and started to try and map where a whole range of different binding sites lie on this plot to predict or explain their selectivity. And this in principle works quite well, but there's a couple of assumptions that I'll come to in a moment. We don't have to just look at sodium and potassium. So here I'm showing that whole range of monovalent alkali cations, and you can see that there's different regions of chemical space that favor different ion types. And this essentially recaptures the kind of selectivity sequences that Eisenman predicted 60 odd years ago. Okay, so far I've talked about 6 different mechanisms for generating selection at a particular binding site. There's the background dehydration energy differences. There's 3 effects that relate to the shape and structure of the pore, and there's 2 effects that relate to sort of the chemical nature of that pore. Now, I think these are good ways of understanding thermodynamic selectivity at a binding site, but there's 2 simplifications that may limit using them on their own. And that is that so far we've essentially assumed that competing ions bind at exactly the same location. Now, if they bind at different locations, then we have to consider how these factors change at those 2 different sites. And also, we've just looked at single ions. We haven't looked at how nearby ions influence binding. And we know that a lot of channels and nanopores do have multiple ions in them. So let's think about how we can tackle these factors. So mechanism 7 is going to relate to this idea of different binding sites. And before I come to exactly what I mean, I just want to present some evidence that ions really do bind at different sites in biological pores. So here we are again looking at the structure of the selectivity filter of the potassium channel. And these are now X-ray structures from Kriener's group in different conditions. But my main point that I want to show is that these structures show these classic potassium binding sites between the planes of carbonyl groups. But they also obtain structures in the absence of potassium and most clearly in the presence of lithium. And from those, what you can infer is that lithium, and sodium most likely as well, binds at a completely different location. It likes to bind in the plane of those carbonyl groups. This picture on the right here sort of zooms in on that. So you can see this green representing the lithium ion binding in the plane of the carbonyl groups with water each side, whereas potassium likes to bind between these planes. So clearly there's some good evidence, I think, that competing ions can bind at quite different sites. So how can we think about this and how to take into account these different sites when looking at ion selectivity? So one way to do that is to come back to this concept of free energy profiles to move an ion through the pore. And so far what we've looked at in those first mechanisms is really the thermodynamics at a particular site. So we might think that there's barriers and there's wells to move an ion through a pore, but all those thermodynamic mechanisms were really comparing the depths of the energy wells at a particular binding site, a minima, assuming that both ions like to bind there. Now, what this more later data is showing is that the ions may bind at different positions. I haven't showed that very clearly here. And perhaps even more disturbingly, their data showed that the in-plane sites had an even greater selection for lithium over potassium than did the between plane sites, the cavity sites for potassium over lithium. So we can imagine that there's different binding sites, and perhaps lithium or sodium binds even more tightly at its site than potassium does at the places it likes to bind. So how can we account for this? Well, of course, that's only looking at individual sites. So if we look at the profile along the entire pore, we can see that perhaps lithium might have large energetic barriers to reach that site, and it's due to the differences in the barriers, not just the minima, that yields selectivity. And so I think we have to look at all of those thermodynamic factors along the entire length of the pore to get the complete picture. We have to think about the kinetic aspects, the rates to get to the sites, as well as the sites themselves, to fully understand these processes. Okay, so we talked about the first 6 mechanisms. I've introduced the 7th mechanism, which I'm talking about kinetics, the barriers along the length of the pore. And I want to just finish up because I know we're getting towards time, and the last one's quite complex, and that's to look at the competition of multiple ions inside the pore. So to introduce this, because I think it's a very important concept, I want to go again to a more clear-cut example. And so instead of looking at potassium channels, I want to look at calcium channels and the selection between sodium and calcium ions. Now, many of you might be quite familiar with this behavior, which is called the anomalous mole fraction effect, which simply says the current through the channel changes as you change the composition of electrolytes on each side. So if we start with just a solution with pure sodium, or should I say, or no calcium, sodium chloride solution, you find quite a large current passing through the pore. That's shown up here. But as you slowly add more and more calcium ions, what you find is that the current, first of all, decreases, close to 0 in this case, before increasing again. Now you can work out that the current carried on this left side of this graph is actually carried by sodium ions. So what this is saying is sodium ions can conduct through the calcium channel in the absence of calcium. The current carried on the right-hand side is carried by calcium ions. So once you get sort of a competing mixture, it's only calcium ions that conduct. In fact, you only need a very small number of calcium ions in your solution to have effectively blocked sodium and be on this right-hand uphill side of the graph. Now, I think we can only understand and explain this using multi-ion effect, because it's very hard if you're looking at an individual ion on its own. So how can we do that? Well, let's think about a calcium channel. Calcium channels, again, have a relatively narrow selectivity filter that stops ions from passing one another, but it's also quite negatively charged. Calcium channels have a characteristic ring of glutamate residues in that selectivity filter. What that does, of course, is attract cations into the pore. So if we just have a sodium chloride solution, the sodium ions are attracted into the pore, and on average, we think you get around 2 sodium ions that are always resident inside that pore. They're just attracted in there by that strong negative charge. And if you want to have conduction, you need to have a knock-on mechanism where another ion comes in and kind of displaces them, moves them on out of the pore. If you have just calcium chloride, calcium is the only cations. Calcium is a divalent charge, so rather than 2 sodium ions, you probably only need to have 1 calcium ion sort of resident in the channel to neutralize the charge. And now it's a second calcium ion that comes on and knocks that first calcium ion through the pore. Now, what about in the case where you have a mixture of ions? Well, this is where the ion competition comes into effect. So essentially, what I'm trying to suggest here is that if calcium gets into the pore, then sodium cannot dislodge that calcium. Calcium is blocking the sodium current. And why is that? That's because calcium is attracted to the negative charge on the protein, and that's being sort of balanced by the repulsion between the permeating ions. But the repulsion between the monovalent and the divalent is less than the repulsion between the 2 divalent ions. And so the sodium basically can't push hard enough to push the resident calcium out of the way. But this kind of explanation only makes sense in a multi-ion pore. You couldn't get this kind of selectivity with just 1 ion passing through at a time. So my reason for introducing this was really just to stress how the competition between multiple ions can be critical to generating selective transport. Okay, now I want to show that this multi-ion competition is probably very important in potassium channels as well. And we all know that potassium channels involve a multi-ion permeation mechanism, so this probably makes sense. But I think the clearest data comes from X-ray structures from Young's group, where they obtain structures not only of classic potassium channels, but a range of other similar channels too. So the classic potassium channel, as we've said, has 4 of these cavity sites for potassium ions between rings of carbonyl groups. There's a very similar naturally occurring channel known as the NAK channel. It has a slightly different sequence that allows some of these carbonyls to swing out. And a consequence of that is that there's only 2 of those classic sites left in the selectivity filter. So this is what they describe as a 2-site channel, and it's actually non-selective for sodium and potassium. Then they were quite cleverly mutated this channel to reintroduce first a 3rd site and then a 4th site back into the channel. And what they find is the 3-site channel is non-selective, but the 4-site channel is selective again. And so somehow the presence of the 4 sites was important. And I think the reason for that is a multi-ion competition effect yielding selectivity. And I've done my best to try and characterize that in a simple fashion here. So why are 4 sites important for generating selection? Well, in a 4-site channel, it's not the case that there's always 4 potassium ions inside that pore. It probably is alternating between 2 to 3 ions. Now, I don't have time to go into this, but there's a whole debate about whether ions pass through with a water molecule in between them, or whether they can actually occupy adjacent sites and knock one another on that way. But for what I'm saying, it doesn't really matter. In either case, you expect at least 2 ions in the pore at any time, and it's probably conduction is alternating between 2 and 3 ions inside the selectivity filter. In a 3-site channel, however, you're probably alternating between 1 and 2 potassium ions in the pore. You probably have like 2 here and 1 here to get permeation, conduction. Why is that important for selectivity? Well, I think it's this. If sodium tries to pass through a 4-site channel, it's going to find either 2 or 3 potassium ions in the way. Now, if it wants to move through, depending on how it finds them, it might be able to find one of these in-plane sites. But essentially, it has to push 2 potassium ions out of the way. And furthermore, the sites where it likes to bind are very, very close, like this, overlapping almost with the potassium. So it has to be able to push them out of the way at a distance because it doesn't like to be in the cavity sites in order to knock them on. And effectively, that outer ion is sort of locking the inner potassium ion in. So there's kind of a lock-in effect that prevents sodium from pushing them out. In a 3-site channel, the sodium has a chance to encounter the channel with only 1 potassium present. And now there's no lock-in effect. And I think it's just easier for that sodium ion to be able to move that potassium between cavity sites while it occupies in-plane sites. There's no lock-in effect. And so here again, it's a multi-ion effect that's important. Selectivity comes from the fact that there's always at least 2 potassium ions in the channel at once. Okay, so we're getting close to the end. So in summary, what I've tried to do is introduce what I think of as 8 different physical mechanisms that can yield selective transport. And I'm actually really interested to hear if you think I've missed any different mechanisms, or I should be breaking some of these up into more than one. Now, as I said, the reason for introducing these is kind of to have a toolkit for understanding selection in a range of different situations. And of course, it's possible that more than one of these are at play in any particular case. Now, given that I've talked a lot about potassium channels, I can't help but sort of add some commentary on what I think is really most important, because all of these 8 mechanisms have been proposed to be at play inside potassium channels. And maybe this will get a little bit of debate going. But my feeling is that we can probably rule out 1 or 2 of these. So the very strict rigid cavity model, I think we can rule out. There's quite a lot of evidence that that the protein has enough flexibility that strict interpretation doesn't hold. And this is somewhat ironic because the rigid cavity or the snug fit model is still the one that you're most likely to encounter in a textbook. What I do think is happening is that it's actually the combination of all of these things. So that competition explanation I gave you at the end relied on kinetic factors, and the kinetics in turn relied on sort of the thermodynamics at all the different positions throughout the pore. So it's actually a combination of all these different effects. And it's much harder to describe that in a pithy sentence or 2 in a textbook than it is to describe a classic snug fit or rigid cavity model. So I think that's one of the reasons that it's been very difficult to offer simple, like catchy explanation for selection in potassium channels. In other cases, it may be more straightforward. There's less of these mechanisms are at play. Okay, so I pretty much said everything I wanted to say. Thanks a lot for your attention. And I do want to acknowledge people that have worked on this. So this is my current group over here. Over a long time, and people that were particularly involved included Michael, Dylan, Chen, and Wanho, and I want to thank them in particular. And thank you all, of course, for your attention. Thank you.
Thank you very much, Ben, for this very clear and interesting presentation and for the perfect timing. So we have some questions on the chat, so I'm going to read the question. The first one is from Mamar Bukabcha, who asks, what is the difference of forces between ions and proteins when you categorize the different mechanisms?
What is the difference in forces? So when I'm categorizing those different mechanisms, I mean, the forces are the same. It's just which one, the balance. Let me think about this. Actually, it's a good question because it's harder to answer than I initially thought. So in all cases, what we're doing is we're looking at the attraction of the ion to the pore, and that comes from electrostatic interactions between the different charges and van der Waals interactions, and the repulsions between the different ligands and water molecules around the ion. So again, that's electrostatics and van der Waals interactions. And it's the balance of all those factors. And the different mechanisms all rely on those same forces, but it's just, I guess, which of those forces we're focusing on. The rigid cavity, the strained cavity could include some of the covalent bonds in the protein scaffold and things as well. But most of these forces, it is still the electrostatic and van der Waals, but it's between which components of the system we're looking at, I guess, as to which mechanism we're talking about.
Okay. And we have another question from Mauro Chinappi, who says, nice talk concerning confinement. You mentioned that for large pore diameter, no selectivity is observed. I was wondering which is the size of the pore so that the bulk behavior is expected, and if pore hydrophobicity may affect the selectivity for pores out of the single file regime.
Yes. So this was when I was talking about the dehydration differences, and I was saying that above a certain radius, the ions can fit through in a hydrated manner, and you don't get much selectivity. Now, pore hydrophobicity will matter in that, because if you have a very hydrophobic pore, you're going to make it easier to dehydrate, or it's going to be more likely to dehydrate ions. If you have a polar pore, it's going to be easier to pull. Some of those water molecules through as well. So as I described it, I was really ignoring ion-pore interactions, but it's a very good question that you've asked, because I didn't talk at all about the water-pore interactions and how they can affect that situation. And to be honest, that's something I've never tried to quantify, but I do think the hydrophobicity of the pore is going to affect the ease of dehydration, and that's obviously going to change the balance leading to selectivity. So it's something to think more about.
Okay, we have some other questions. One is from Luigi Catacuzzeno about mechanism 8. It is not possible that calcium is preferred to sodium for entropic reasons, because one calcium has more free volume than two sodium ions inside the pore?
Yes, that's a good suggestion as well. And I didn't go through all the different mechanisms suggested for selectivity in calcium channels, but that has been one of them. That essentially, yeah, calcium has a charge of +2, but a smaller volume than 2 sodium ions. So that is plausible. I agree with you. Now the question comes down to whether the entropic consideration, the volume, is the limited volume of the pore important or not? I have my own personal view here that it's, you know, it's a relatively large pore. I don't think that entropic contribution is most significant, but there are others that would disagree with me. But regardless of whether it's the electrostatic attraction or the entropic difference, I still don't think you'll be able to get the degree of selectivity we've talked about if there's only a single ion present. I mean, by definition, that entropic explanation is comparing 2 sodium ions versus 1 calcium ion, and the permeation is going to involve another ion to come and replace that one. So even with that one, we're still talking about 2 to 3 ions involved in the conduction process. And I still would probably classify that as a multi-ion mechanism, but it's a good question because you've really pulled up some of the subtleties of that example.
Okay, and about it, there is a follow-up comment from Roland Roth saying, there are studies on selectivity that confirm that entropy can play an important role. Okay. And then we have the last question. We have still 2 minutes. Impressive talk and clarity. How is the chemical nature of the ligands quantified in the phase diagrams plot? And the question is from Antonio Tinti.
Okay, yes, so those phase diagram plots are calculated in sort of simple models. So there's different ways of doing it. The chemical nature of the ligands will give you selection between different solvents. So computationally, we can do it, for example, by taking a box of water, a box of another solvent, formaldehyde or something else like that, and you can calculate the partitioning between them, or the free energy to move the ions, to swap the ions from one solvent to another. So that's one sort of way of doing it. Of course, computationally, we're not constrained by specific chemical groups. We can artificially change the partial charges and the dipole moments on the different solvents. And so the way those kinds of phase diagrams were done was to take sort of an abstract dipolar ligand and to artificially change the dipole moment by changing the partial charges, and to step through a range of different charges to see how that affects the free energy to swap the ions between different kinds of solvents. And in that case, we were always using the reference as water. So it's always imagining we're coming from an aqueous water solution into a ligands that are ligands of the specified dipole moment. I hope that answers the question.
Okay, so thank you again, Professor Corry.